Okay, so now we're recording. We'll go ahead and start. Remember, this week is covering mainly chapter 8 and chapter 13. Actually, let me do that off. So we're mainly doing chapters 8 and 13, but remember for chapter 8, there's also a small section that is actually in chapter 9. So the section on horizontal gene transfer is in 9 and not 8. So the learning outcomes that you guys have for these chapters are here on the first slide, just to help keep you organized. So as we move through the genetics section, some of this should be review for people from things like general biology, but a lot of it depends on which biology class you took as to what level you got for a lot of the molecular processes. As a note, in the exam module, all of my biology 1610 lecture videos are there. So if you want some other details or you want to see slightly different lectures, kind of in the same style that I do with this class, they actually are on Canvas that you guys can use. So let's go ahead and start looking at the DNA replication process. So everybody should be comfortable with the enzymes that are involved in DNA replication and kind of what their job is and what order they're working in. So I just have the enzymes on. I'll point out the single-stranded binding protein since we had the question at the start of this on them, but since they're not an enzyme, they're not actually listed on here right now. So if we were to put these enzymes in the correct order, replication starts kind of with both DNA helicase and a topoisomerase. So DNA helicase is what binds to the origin of replication, starts unwinding the double helix, but as it's unwinding that double helix, it's going to need to use a topoisomerase in front of it, which relaxes some of the supercoils. And what supercoils are is if you take a rubber band and you kind of rub one end between your fingers, you'll notice the other end is going to wind up. That's what supercoils are. Because bacterial DNA is round, that's happening as we're unwinding it. So topoisomerase just needs to remove all those coils happening at the other end. As we start unwinding, remember there are single-stranded binding proteins that will bind to keep everything from coming back together. But then the next protein that we need as an enzyme is going to be RNA primase. That comes in and puts your small RNA primer, so the little short sequence of RNA, and is needed because we can't just stick DNA into nothing. It actually has to have a 3' OH group to attach DNA nucleotides to, and that's what our little RNA primer gives us. The next step is to make the new strands of DNA, and the main workhorse enzyme for that is DNA polymerase III. After it's done its job and made all the strands that it can, the next enzyme is DNA polymerase I, which is going to remove those pieces of RNA and replace them with DNA. And then we're going to have your ligase that's going to come in and join your little Okazaki fragments together that were found on that lagging strand. And then we actually come back around to our topoisomerase, because at the end of all of this, when you look at DNA replication in bacteria, you actually ended up with one double-stranded molecule of DNA that's a circle, and then the other strand that you've been making is also a circle, but they're stuck together. So this one runs underneath the blue one. And so topoisomerase at the end is actually going to cut this and separate them. So we end up with one completely set genome and then the other completely set genome. So those are the main enzymes. You should have a sense of how they function. I'm going to ask the group, does anyone want to see me diagram and draw out replication and actually see the process? Or are you comfortable just refreshing what those enzymes do? Anyone with questions on DNA replication? Okay, doesn't look like it. So we'll jump to our next set of molecular processes, which are really looking at transcription followed by translation. So DNA replication is needed for when bacteria are growing, when they're going through binary fission and dividing, we need to replicate so we can pass that DNA to the next generation. For the transcription and the translation, this is how we're getting the information that's stored in DNA into functional information in proteins. Because remember, DNA just kind of sits there. It's the storage, it's the proteins that are really doing things. So this question says, in bacteria, the sigma factor associates with the promoter, similarly to how the small ribosomal subunit associates with what? So go ahead in the chat, What do you guys think the answer to that one is? A, B, C, or D? Okay, so I'm seeing a couple C's show up on there. That is the correct answer. So as we look at the process of transcription and translation, we start with DNA. So here's our 5' and our 3' ends of the DNA. And somewhere in that DNA is an actual sequence, so a specific spelling of the nucleotides that is the promoter. And that promoter, we typically say, has a -10 and a -35 region. That's where the key spellings are. What binds to those regions was the sigma factor. So this is the sigma factor, which then, remember, allows your RNA polymerase to bind, and it's the RNA polymerase that releases and actually does transcription. So from here, we do transcription. We get our messenger RNA. And remember, it's got a 5' end and a 3' end. And then somewhere in there's also a specific spelling that is the Shine-Delgarno sequence. And that's where the small ribosomal subunit is going to bind. It scans to find the magic codon. Remember that AUG. The rest of the ribosome joins it, and then we get translation into a protein. So this is a very controlled process. Promoters identify where the start of genes are in the DNA, and then the Shine-Delgarno sequences identify where the translation needs to begin in the messenger RNA itself. Also remember that these Shine-Delgarno sequences, these are what allow codons to exist in bacteria where they can't exist in a eukaryotic cell. Because in a eukaryotic cell, each of the messenger RNAs gets a 5' cap, and that's actually the ribosomal binding site. So in a eukaryote, it's impossible for the ribosome to bind somewhere over here in the middle of the DNA. It has to bind at the front end. With Shine-Delgarno sequences, we could have another Shine-Delgarno sequence over here, and a ribosome could bind here, so there could be more genes on there. And that's why you can get operons in your bacteria, but not in the eukaryotic cell. We're going to continue looking at these transcription translation processes. So I'll move to the next slide before I stop to see if we have any questions. Okay, so during that translation, ribosome comes in. It's going to actually be joining the amino acids together. But in order to know what amino acids to join, we need these tRNA molecules. So these are the ones that are bringing the amino acids to the ribosome so that the ribosome can join. And remember, they know which amino acid to use because their anticodon region, which is down here at the bottom of the tRNA molecule, is going to be used to read the messenger RNA itself. So we've got codons that are in the messenger RNA, okay, that are read by the anticodon of the tRNA. And that specific anticodon would be attached to a key amino acid, okay, that would be on the 3' end. So if we look at these, and the question wants to know which of these 4 tRNAs would be used to translate the codon CGA in the messenger RNA. So go ahead in the chat, type in what you think A, B, C, or D. If you guys can't read the codons, I'll write them a little bit bigger here. This one reads TCG. This one is GCU, UCG, and CGA. But the key to keep in mind when you're answering this question is to pay attention to where your 3' and 5' ends are. So when we read these, this is the 3' end, this is the 5' end of each one of these molecules. Okay, so I'm seeing a lot of B's show up. And I specifically did this question because it is a little bit of a trick question. So it's actually not B. In this case, it is actually C. And this is why. Whenever you have complementary base pairs, so when C binds with G or G binds with C, or A binds with U, so on and so forth, the strands have to be anti-parallel. So where you have the 5' CGA codon, you would have to have a 3' GCU anticodon. And so this is that anti-parallel nature. Okay? These are opposite ends of the strands so that we can get our C's and G's and A's and U's. But what that means is that this sequence is really 5' UCG 3'. Okay, so some of the trick with looking at nucleotides and kind of how they function is that this sequence, 5' UCG, is the same exact sequence as 3' GCU. We're just not used to reading backwards a lot of times, and so we rearrange it to fit what we are used to seeing. But that's why it's important to put 5s and 3s, because as long as that's there, these sequences are equivalent, they're identical. Okay, so everyone got the concept that the complementary base pairs are forming between the anticodon and the codon. It's just keeping track of what direction is that anticodon going to be lining up with the codon itself. And then at the top of all of those tRNAs would have to be the proper amino acid. And so for this one, this anticodon region that would line up with our CGA codon, if we look at a codon chart and we find CGA in here somewhere, so here's CGA, this is an arginine amino acid. So if we came back up to this slide, what would be attached here is an arginine. So that's how the tRNAs are able to be the codon chart of the cell, is they say, what's the codon in the messenger RNA? It's going to be tied specifically to that amino acid. Whereas we luckily have the codon chart, which makes our life a lot easier when we go to do these types of problems. So this question is going to ask us to identify both the DNA and protein mutations that are occurring in this gene. In order to do that, first you need to be able to translate. And so for this class, I will always give you the messenger RNA sequence. For my 1610 class, I make them do the transcription. There's enough information in these 2 weeks that I don't need you guys to kind of relearn or understand how to transcribe. You need to understand the process and the kind of sigma factor and what's involved, but I won't actually ask you to do transcription. But you do need to be able to do translation. So remember, if we were going to translate this, there's a Shine-Delgarno sequence somewhere in here, so that the small ribosome could come in and bind. And what it's looking for is that AUG. And you need to find the AUG to set your reading frame. And then that sets your group of 3 codons. Then to figure out what the protein sequence is, we're just going to look in the codon chart. AUG is always methionine. And now we've got a CUC that comes next. And so we say, okay, here's CUC, that's a leucine. GAU, they say, okay, where is GAU? Here's GAU, it's asparagine. And then we get to UGA, and you'll see UGA is one of the stop codons. So we've reached the end of our protein. We just have a little 3 amino acid protein here. Now we want to look at the next mutated sequence to see how they're different. And so you get kind of used to reading C's and G's and A's and T's when you do this type of work. So we're going to scan across and say, okay, C, C, A, U, G, those are the same. And then we have a U here. And if we look up here, there was a C. So the question is, did that C become a U? That would be a point mutation. Or did we lose the C? And so if we keep going, notice here we've got a U, and then we've got a C, a G, an A. So this sequence that continues is the same as this sequence, which tells us we really just lost that nucleotide. And that would be a deletion in the DNA itself. At that point, you might not even need to do the translation, because remember, deletions and insertions, as long as they're not 3 nucleotides together, will always lead to a frame shift. But let's look at the translation anyway, just to see. So here's our AUG that gives us the methionine. Now we've got a UCG. So if we come back here, we find UCG is serine. And we've got AUU. AUU is isoleucine. GAC is asparagine. And notice we haven't hit a stop codon. So if this sequence kept going, our codons would keep going and we would have other amino acids here. So when you look at this and we say, okay, we went from methionine to a serine, a lysoleucine, and all of those downstream had changed, a frame shift deletion, because we've moved our entire reading. Okay, so I'm going to pause there for a minute. Does anyone have, whoops, that was not what I meant to do. Let's go back up to where we were. Okay. Does anyone have questions on transcription or translation, either the processes or how to do them? Okay, we'll move on to the next part then, which is the gene regulation. So gene regulation is kind of my baby. This is what I actually study. I study gene regulation in tuberculosis, which is really looking to see how it expresses its proteins when it's in the human body compared to when it's not. Because if you can figure out how that gene expression changes, you start to identify things that might be drug targets. So I love gene expression. I could do an entire class on it, but we won't. We will do one short little piece on it. So I'm going to give you guys a minute to read through this question because I do want you to try to answer it on your own. So when we look, we're saying, okay, we're working with a mutant E. coli. Okay, so there's some mutation in this E. coli strain, and that mutation is in the LAC-I protein. What the mutation does is it makes it unable to bind to allolactose. So the question wants to know, what is that going to mean for E. coli's ability to actually use lactose for energy? A says that E. coli is always going to be able to use lactose, even when glucose is there, because if the LAC-I protein can't bind lactose, it will never be able to bind the DNA to turn off transcription. Next one is saying, okay, E. coli will be able to use the lactose, but it's going to do so slowly. And this is because the LAC-I is an activator protein, but if it can't bind lactose, it can't bind DNA. And then C says it will never be able to use it because the LAC-I protein is going to be permanently bound to the operon, repressing transcription because it needs to bind lactose to be removed from the DNA. So which one do you guys think, A, B, or C? Okay, so I see a couple C's up there and a couple of B's. The answer to this one is C. And so this is one that students typically have questions on. The key with the key with B here is keep in mind LAC-I. I stands for inhibition. So the LAC-I protein is a repressor protein, not an activator protein. So this is C because the way this operon works is remember we've got the DNA, the genes of the operon are down here. So these are the actual LAC operon genes, but we really care about the regulatory regions. So we've got what's called an operator site. We've got the promoter. And we've got the activator binding site. And so what's normally happening is the LAC-I protein, because it's a repressor proteins always bind to operator sites, and they physically block RNA polymerase. So RNA polymerase wants to bind to the promoter to do transcription, but with LAC-I in the way it can't, and so this is off. The only way we can go from off to on, that does not say on, from off to on, which would then allow our transcription, is if we remove that LAC-I. And the way that works is if lactose shows up in the cell, which means lactose is now in the environment, okay, so this is what we call allolactose, which is just a slightly modified version of lactose. It's going to come in, it's going to bind to the lac-I, and when that happens, the lac-I changes shape. So our lac-I changes conformation, and it can no longer bind the DNA. It's removed from the whole process. Put our lactose on there. And when we remove the lac-I, now RNA polymerase is able to get through and do the transcription, we turn it on. So that's what's supposed to happen. If we go back and we look at the problem itself, the problem said that in our mutant, the lac-I protein can no longer bind allolactose. So when we come and say what was supposed to happen, we're looking here, if this step can't happen anymore, then we're never able to change conformation and pull it off, which means lac-I will always be bound to the operator, and this will always be off, even if there's lactose around. So even if the cell wanted to use that energy, use it for energy, and needed to break down the lactose, this mutation wouldn't allow it to. This tends to be how I like to ask these types of problems, because you have to do the thought process of what normally works, and then how does that change with the mutation itself. The other key to keep in mind with the lac operon is that there's a whole other level of regulation to it. So we always pay attention to the repressor first, so whether or not lactose is present or absent, because if lac-I is here, it doesn't matter what's happening with the activator protein, it's always going to be off. That lac-I is always in the way. But if we have removed it, so remember we've got the operator, the promoter, the activator binding site, and say in this scenario, lactose is present. And so our lac-I, remember, has been removed from the DNA. It's bound to lactose here. And so it's not in the way anymore. In this scenario, now we care about whether or not glucose is around or not. The glucose that we care about, remember, we learned about catabolism, we learned about glycolysis, where glucose is what starts that whole process off. So if glucose is there, the cell wants to use it because it's easier. Bacteria want to save as much energy as they can. It's the whole purpose of gene regulation is to conserve energy. So if there's glucose, the cell wants to use that, doesn't really want to use the lactose. So in this scenario right now, we'd have a little bit of RNA polymerase binding, we'd get a little bit of transcription, and so this is on, but we're getting low levels of expression. But the cell's using up the glucose. Now eventually it will run out and glucose will decrease. What happens in the cell is when there's glucose present, we have low levels of a molecule called cyclic AMP. This is actually what I study in my lab. As the glucose goes away, the amount of cyclic AMP in the cell is going to increase. So now we've got little blue cyclic AMPs that are floating around in the cell. What that allows is going to be the opposite of what happened with the lac-I. So there's an activator protein called CRP, for cyclic AMP response protein. This is the activator, but when glucose around and there's no cyclic AMP, just float in the cytoplasm. Once cyclic AMP shows up, cyclic AMP binds. That again changes conformation, but now we change to a conformation that can bind to the DNA. The activator protein binds the activator binding site, and the whole purpose of activator proteins is to recruit RNA polymerase. So now we get a lot more RNA polymerase coming and binding with the promoter, remember, along with the sigma factor. This means we're now going to get lots and lots and lots of transcription, which means we're going to go from low to now high levels of expression, because we were low when glucose was present, but we want high expression when glucose is absent, because if there's no more glucose to feed into glycolysis, we're going to need to use that lactose sugar to get energy. So we need to get high levels of expression, because the whole purpose of the genes of the lac operon are to break down lactose for energy. And so as you look at gene regulation, you really want to pay attention to why things are regulated, and that will help understand, are there going to be, what the expression levels are going to be. So are there questions about the gene regulation sections? Lac operon seems to make sense to everyone in the concept of activator proteins and repressor proteins. Okay, if there's no questions, we'll move on to the very last question then for this genetic section. This is one, again, remember, I like to ask questions that aren't just what's the definition of something. So this is a way to ask about horizontal gene transfer where you need to understand the different mechanisms. So we talk about horizontal gene transfer, we're talking about conjugation, transformation, or transduction. And you need to understand what those are to be able to answer this question, but it doesn't just ask you for a definition. So if we take a look at the question itself, we're trying to study horizontal gene transfer mechanism in species. So we know it does horizontal gene transfer. We're trying to identify how it might do that. So what we've done is we've put a donor on one side of a filter and a recipient strain on the other. What that typically looks like is in your broth container, you've got a filter in the middle, and we would have the donor on one side and the recipient growing on the other side. That filter has pores that are about 500 nanometers in size. When we look at this, we're saying DNA transfer has not actually happened. So read through A, B, C, and D and decide what you think the answer to the question is. Go ahead, put it in the chat. Okay, so A is saying that species have B is again saying they have to use conjugation, but then they're saying that species must be naturally competent with that DNA binding protein. So go ahead, take some guesses. Okay, seeing some. The answer to this one is A. Okay, so this is conjugation. The reason it is conjugation is this filter with a pore size of about 500 nanometers would allow viruses to get through. Okay? So viruses are typically smaller than that. So viruses would be able to go back and forth here. It's also definitely small enough to let DNA go through. And so because DNA transfer didn't occur, the only one that's left there is conjugation. Remember, conjugation requires cell-to-cell contact, and your bacterial cells cannot go back and forth, which means it must then be conjugation to do. Okay, so before we move to the next chapter, does anyone have questions on horizontal gene transfer or on any of the genetic concepts?
I actually was wondering if you wouldn't mind going over this horizontal transfer mechanism a little bit more, because I'm still a little bit lost on that one.
For just conjugation or for all three of them?
For all three, if you don't mind.
Okay. Yep.
Thank you.
Okay, so remember, overall, the concept of horizontal gene transfer is just a way that two different bacterial cells, okay, so we've got DNA in one, this DNA can somehow get from this bacterial cell into this bacteria. And there's three ways that can happen. For transformation, actually we'll do this. Make our donor blue. So for transformation, what happens is at some point that cell actually dies. When it dies, it's going to start to fall apart, it's going to start to decompose, and its genome is going to get chopped up into little bits. For whatever this species is, if it's able to do transformation, what that means is the cells have to be naturally competent. So on their surface, they have proteins that can do DNA binding. Okay, they're able to bind to DNA. When we say that they're competent, that's all we mean, is that they have those DNA binding proteins. Because what will happen is the DNA that's now just floating around in the environment, this is what we call naked DNA, is going to come over, it's going to bind to that DNA binding protein. And when that happens, that signals the cell that says, hey, there's DNA out here, bring that into the cell itself. That's all transformation is it's taking that DNA floating around in the environment, allowing it to bind to the cell surface, and then the cell pulls it in. Keep in mind, though, that because this is linear, now that it's in the cell, if we want it to actually stay, it's going to have to do recombination, which means it's going to have to become part of the chromosome itself. And so recombination just means that this piece of DNA swaps in to the chromosome itself. Okay, so that's transformation. And Jason, I see your question in the chat asking about what competent cells are again. Competent cells, when we talk about cells that are competent, means that they can do this process. So they can do transformation, because not all bacterial species can. And so cells that are able to do transformation can do it because they have these DNA binding proteins on their surface. So that's what we mean by naturally competent, is that the cell itself produces a DNA binding protein, it's on the surface, so it can bind that naked DNA and do this whole process of transformation. I tend to say naturally competent because in the laboratory, we can take cells that cannot do transformation and force them to. So you can actually mix them with certain chemicals to make competent cells, and then they'll do transformation just fine, because it's really this binding part that's key to the whole process. That's kind of the difference. It's why I typically will say naturally. We use this in molecular biology and microbiology all the time to make bacteria take up pieces of artificial DNA for us. Okay, so does transformation make sense before I move on to the next one?
I just had a curiosity question. So with the DNA that gets replaced with the recombination, what happens with that older DNA that's no longer part of the circular genome?
That's a good question. So if we actually draw recombination kind of bigger so that we can see it. So here's our DNA that's coming in. This process, keep in mind, is typically happening because of what we call homologous recombination. We just kind of leave the homologous off, which means some of the sequence has to match. So like if there's an ATTG there, there's also going to be an ATTG there, and same thing over on this end, a CCG. So when I draw those little X's, that's actually what's happening, is this is matching up with here, and this is matching up with here. And then they literally swap spots. So as we were traveling along the genome, we're going to jump from here up to this piece of DNA, travel down here, jump back down to this one. And so they do literally swap places, where now that's been cut out and it's been replaced with this one. And now that this is the new linear piece of DNA, in bacterial cells, linear pieces of DNA are degraded, so it's just going to get broken down. So typically either the new DNA would get degraded if recombination didn't happen, or the old DNA gets degraded when it gets kicked out of the genome. Any other questions before I move on? Okay, so transduction is when we're using those bacteriophage. So remember, this is the one that the bacteria aren't actually doing anything. They just kind of get lucky and they're like, oh, well, I gained a new piece of DNA instead of being infected by a virus. Because what happens for transduction is, here's again the cell that's going to end up being the donor cell. So when this cell gets infected by our bacteria, that cell starts making all sorts of little bacteriophage. When we talked about viral replication and assembly, there should be viral DNA in the heads of all of these. But what happens is that during this process, a lot of times the bacterial genome will actually get degraded, so they'll form lots of little pieces. The virus forces this to happen so that all the cell's energy can be spent on the virus. And when that happens, every once in a while, these little pieces of bacterial genome end up packaged in the virus instead. And that's now a problem. So when this cell lyses and releases this little bacteriophage that now has bacterial DNA instead of viral DNA, now it's not going to cause an infection anymore. Because remember, your viruses are mainly just little genome carriers. And so when we come over now to who are as our donor, it gets infected by this specific phage. Instead of injecting phage DNA, we inject that new DNA, and then again, recombination can happen to get into the genome itself. So completely by accident, this cell has now gained some new DNA just because the virus made a mistake. Questions on transduction, or does that one seem to make sense? Okay, and then the last one is conjugation. This is the one that's typically referred to as cell sex because they do have to have cell-to-cell contact. And conjugation's a little bit more specified. So in conjugation, we have two cell types, cells that are considered to be F positive and cells that are considered to be F negative. And that's based on the presence or the absence of what we call the F plasmid. The F plasmid has the genes necessary for conjugation. So that means the only organisms that can initiate conjugation are ones that have one of those plasmids. And they're always going to initiate it with cells that don't. So these are always going to be your donor, and these are going to be your recipient. When conjugation's initiated, the F positive cell reaches out and forms what we call the sex pilus with the F negative cell. And this F plasmid is going to do what we call rolling circle replication. So if I draw it more correct, remember DNA is double-stranded. And so what ends up happening is that you actually nick this outside strand, and then you drag the outside strand into this new cell. What that ends up doing is we remove the outside strand because it's now in the donor, moved from the donor to the recipient, and then DNA replication has to happen to go ahead and rebuild the double-stranded DNA molecule in both this cell and this cell that got the one single strand. And what's happened is our F negative is now an F positive cell. Conjugation can get a little bit more complicated with the HFR cells. So an HFR cell is a cell that the F plasmid actually went through recombination and is no longer a plasmid, but is now part of the chromosome. And so what happens here is it still has all of those genes for conjugation. It can still go ahead and initiate conjugation, so we can still form that pilus with an F negative cell. But now, remember again, we are technically a double-stranded molecule through here. Now when we nick this strand and we go to transport it, we're going to get some of that F plasmid, but we're actually going to drag all of the rest of the chromosome here along with it. The problem is that's not very stable. Eventually this is going to break and you never actually transfer the whole thing. So the HFR cells are special because you don't just get the F plasmid, you're also going to get whatever extra genes were on that chromosome. But because we don't get the whole thing, this is not a circle, and so it is going to have to do that recombination into the genome to actually stay there. That's why we call them high frequency of recombination, is because the recombination has to happen. Okay. Does that help? Other questions about horizontal gene transfer?
I have a question on recombination, if that's okay.
Yep.
So, for example, if we look at that HFR cell, the initial part of the genome that the F plasmid that got incorporated into the genome of the cell, it did that through recombination. Is that correct?
Yes.
So, and then at the end of that mechanism, it also would have to do recombination. So my question is, does it matter if the DNA is circular or linear to do recombination?
No, and that's a good question. So plasmids, so circular pieces of DNA can do recombination, but they don't have to. So the bacteria will replicate and use circular pieces of DNA. For linear pieces of DNA, they have to do recombination or else they'll get degraded in the genome. And so any type of DNA is able to do recombination if you put it next to other pieces of DNA that might have some similar sequences. It doesn't really matter the format. It's just that the linear ones are the ones that have to do it or they'll get degraded. Does that make sense?
That's perfect. Thank you.
Okay. Any other questions before we move on to chapter 13? Okay, so these I just gave a couple, and these are really more basic multiple choice questions. Out of all the chapters for this upcoming exam, 13 is probably the easiest because it's less about understanding processes or kind of solving problems and more just thinking about the information and understanding the information. And so this one is a lot more straightforward in what you need to kind of know and understand from it. So we'll just walk through a couple of these multiple choice questions, which mainly are checking to make sure everybody understands the overall terminology that we're using in the chapter. Okay, so you're in charge of purchasing some chemicals that you're going to use to disinfect the surfaces around your clinic. I want to know, what are you going to have to consider to make your decision? So A is saying, all right, we're going to need to disinfect the surfaces, so we have to find something that will remove all living things, including spores and viruses. B says, we're going to have to do it, so we're going to have to find something with a small d value that will kill most species. C is saying we're going to find something that's safe to use on human tissue because you're going to be using an antiseptic. And then D says we need something with a large d value because that's going to indicate that it kills many different types of microorganisms. So go ahead in the chat. What do you guys think? A, B, C, or D? Okay, so I'm seeing some B's and some A's show up. The answer to this one is B. So the D value, okay, got to remember what a D value is. That's the amount of time it takes to reduce the population by 90%. So remember that bacterial cells, okay, if we had a population of cells that was, say, this much, and we added our disinfectant, the cells die at a set rate. Okay, that rate is the D value. And so the amount of time that we've decreased exponentially, so from say 10 to the 9 to 10 to the 8, which is 90%. They don't immediately all drop to 0, okay? And so we always want something with a smaller D value because that means they're going to kill the microbes faster. Okay? Large D values means it takes a long time to reduce the population, and that might not be the best. It's not A because we're talking about disinfecting surfaces. And for disinfecting surfaces, we typically are not going to be removing endospores. So when we look at these, if we have to remove all endospores and all viruses, that's the definition of sterilization. And typically the only things that actually get sterilized in healthcare are things that might break into the blood barrier. So in dentistry, it's a lot of the dental tools that if you're going to possibly make your gums bleed, they're known to being sterile. In surgery, it's scalpels and things that are going to be going into the body. But surfaces are typically disinfected, not sterilized, where we're just removing most of the microorganisms. Okay, so kind of key terminology to lose in there. And C, we're not talking about antiseptics. Remember, antiseptics are used on tissue, and they are different than disinfectants. And there are some disinfectants that you could use as antiseptics. Things like ethanol can be typically used on the skin. Yeah, everybody's using ethanol hand sanitizer right now in the middle of the coronavirus pandemic. It can also be used on surfaces. But other things like bleach is typically going to be a disinfectant, but it is not an antiseptic. You're not usually going to dump bleach on you to, clean out a wound because that's going to damage your tissue also. Okay, a little bit more working with terminology. So now we're working to develop a new antibiotic, and we've got two possibilities. Okay. Drug A, we see, has an ED50, so that's the effective dose 50 of 3 micrograms per milliliter, and a TD50, or the toxic dose 50, of 300 micrograms per milliliter. Drug B has an ED50 of 3 micrograms per milliliter also, but a TD50 of 30. So we're looking at these values, and if you're the one working at this pharmaceutical company, we're going to decide which one is going to be the better drug to move forward with looking at stronger. A is saying drug A is the better one because its chemotherapeutic index would be 100. There should be an E there. Compared to drug B's, which has a chemotherapeutic index of 10. A higher chemotherapeutic index means we have less chance of toxic side effects. B is saying drug B for the opposite reason. So its index of 10 times safer than drug A because it's got better selective toxicity. C is saying drug B because the toxic dose 50 is 10 times lower. If it's less toxic, then it's going to be safer. And drug D says drug A because its toxic dose 50 is 10 times higher, indicating that it's 10 times less toxic. So if we look at those options, what do you guys think the answer to this one is? Good. So I'm seeing some A's show up. A is the answer. Remember your chemotherapeutic index, the way you calculate that is it's the TD50 divided by the ED50. Okay, and so for drug A, that would be 300 divided by 3, which is 100. Okay, drug B, that was 30 divided by 3, which was 10. And what that's telling you is how safe is the drug. A higher chemotherapeutic index means more selective toxicity. The drug is harming the pathogen, but it's not harming your own cells. The lower that number gets, the more chance there are of side effects, because the more chances the drug is going to cause toxic effects in your cells itself. Okay, so before we move from the kind of chemical, physical things into antimicrobials, anyone have any questions just on terminology? Okay, so this one's going to say, which of the following explains why there are so many more antibiotics, so things to treat bacteria, than there are antifungals, things to treat fungal infections? Okay. A says it's easier to kill bacteria because they're more simple. B says drugs with high selective toxicity are easier to develop because there's many more targets for antibiotics that are not found in our own human cells compared to fungi because they're eukaryotic. C is that drugs with low selective toxicity are easier to develop for the same reason that was described in B. And then D is that fungi have that chitin cell wall, which makes it challenging to get drugs inside. And if they can't enter, they can't kill them. So what do you guys think in the chat? A, B, C, or D? Good. So this one is B. Okay. This is the whole key with antibiotics is they have high selective toxicities. Bacteria are prokaryotic. They have many more targets in them that are different than our human cells. Remember, the two big ones are the cell wall and the 70S ribosome. So large majorities of our antibiotics target those in different ways. And then there's different metabolic processes, different enzymes and bacteria that we can prevent from working with different drugs. If they don't exist in our human cells, that means we're really not killing our human cells at the same time. And that's the benefit of having antibiotics. And it's why it's harder than making antifungals, or it's harder to make antiparasitics. Those eukaryotic cells just mean they have less targets in the cells themselves. It's also why we don't really have very many broad spectrum ones. You have to actually find specific targets in the specific pathogen, and which makes one drug, say for malaria, not going to work against other eukaryotic parasites that might be causing infections. And the last one is looking at antibiotic resistance. So how do we become resistant to these drugs that we have? And we want to know which one is not a likely mechanism. So A says the bacterial cell has gained a plasmid through transformation. It does horizontal gene transfer. And that plasmid had a gene on it that made a protein that allows for the degradation of the antibiotic. A says the bacterial cell had a spontaneous mutation in its genome that changed the target of the antibiotic so the antibiotic can no longer bind to that protein. C, the bacterial cell gained an F plasmid through conjugation. And on that F plasmid was also a gene for an efflux pump. And that's able to bind and pump various antibiotics out of the cell. And then D, the bacterial cell had an induced mutation in its genome that changed a transport protein. So now it transports high levels of antibiotics into the cell. So which one of these is not a likely mechanism for antibiotic resistance? Good. So I see lots of D's showing up there. So when we talk about antibiotic resistance, there's three main ways bacteria can be resistant to antibiotics. They can either degrade or modify the antibiotic itself so it no longer functions. They can change the amount of antibiotic getting into the cell. And so that either is by preventing it from entering or pumping it out as fast as it comes in, or they can change the target of the antibiotic. So when the antibiotic gets there, it no longer has the ability to bind. And if it can't bind to what it's supposed to be stopping, it can't actually stop it and function. And so those are the three mechanisms that resistance actually functions with. Remember, those resistance mechanisms are gained through either mutations in the genome directly or those horizontal gene transfer mechanisms. You'll notice I didn't have any questions in here asking about what specific antibiotics did. I want you guys to understand what the major target areas are and why those are major target areas, but I'm not asking you to memorize a whole slew of antibiotics and what they specifically function as. Those are the types of things that if you need to know it, it's pretty straightforward to look it up. And if you understand why those are targets, then you understand how the antibiotics themselves function once you have looked them up. So any questions about Chapter 13, remember this is really more informational for that one and just understanding the terminology of kind of how we control microbes and what those various chemicals and antibiotics kind of function as.
I actually just have a question. Do you have those questions in Canvas? The ones that you gave to us today?
Yeah, they weren't there yesterday. I actually typically try to put them there earlier than that, but I forgot about it. But they are up there now, so they went up this morning, which means you can pull them up. They're in the resources, under the resources heading in this week's module.
Perfect, thank you.
Okay, so I stopped sharing my screen. Any other questions from anyone, either about either of these two topics or the upcoming exam, anything about the class in general? Going once, going twice? Okay, if no one has any questions, I'll upload the recording of this later this afternoon once it's processed and saved. And you guys have a good weekend and good luck up on the exam. Bye, everyone.